My dear students, in the last lecture, we have covered biosynthesis of pyrimidines, that is cytosine, uracil, and thiamine, and we have also studied in detail mechanism of action of antineoplastic drugs. Now today we are going to deal with the last portion of this topic. Contents of today's lecture include one of the important disorders associated with biosynthesis of pyrimidines, and that is called Orotic Aciduria. Then we'll see the reactions for the salvage of pyrimidine bases. Afterwards, we would focus on degradation or breakdown of pyrimidine nucleotides. And lastly, we would be focusing on natural and synthetic derivatives that can be obtained from purines and pyrimidines and their significance in health and disease. Orotic aciduria is a genetic condition which prevalence is rare. It is characterized by the deficiency of UMP synthase, and we have seen in our last lecture that UMP synthase is the combination of two enzymes called as orotate phosphoribosyl transferase and OMP decarboxylase. So whenever UMP synthase is missing, it may result into excessive levels of orotic acid in blood, causing orotic acidemia, and also in urine, which is called orotic aciduria. Whenever UMP synthase is missing, then UMP cannot be formed. If UMP cannot be formed, then the other pyrimidines like cytidine monophosphate or thymidine monophosphate and others cannot be formed, and thus pyrimidine biosynthesis is affected. Now, defective pyrimidine synthesis may result in poor growth. It may also result into the development of megaloblastic anemia because red blood cells would be having defective synthesis of DNA due to impaired pyrimidine biosynthesis, and thus red blood cells would be growing in size but won't be dividing, and as a result of which would be resulting into the development of megaloblastic anemia. Types of orotic aciduria. Type 1 orotic aciduria is an autosomal recessive genetic disease. This is characterized by the deficiency of UMP synthase enzyme. And in this disorder, both domains of UMP synthase are defective. For example, orotate phosphoribosyl transferase and OMP decarboxylase. When these two domains of UMP synthase are defected, then orotate cannot be converted into UMP. And there is increase in the concentration of orotate in blood and in urine. If it is present in excessive concentrations in blood, then this is called orotic acidemia, and if in urine, then it is called orotic aciduria. The other clinical manifestations which are associated with type 1 orotic aciduria are growth retardation and megaloblastic anemia, and we have discussed these 2 points in the last slide. Type 2 orotic aciduria is also an autosomal recessive inherited disorder, and in this disorder there is deficiency of only one domain of UMP synthase, and that is OMP decarboxylase. So here you can see that OMP decarboxylase is missing here. So OMP, that is orotidine monophosphate, cannot be converted into UMP. As a result of which there is increase in the levels of OMP and it causes oretidinuria. And when OMP is not further converted into UMP, there is also rise in the levels of orotic acid. So this may also cause orotic aciduria. The further complication associated with type 2 orotic aciduria is megaloblastic anemia. Minor types of orotic aciduria. The very first one is orotic aciduria that is associated with Reye syndrome. In Reye syndrome, mitochondria become damaged or defective, and as a result of which mitochondria are unable to use carbamoyl phosphate. And thus, this carbamoyl phosphate cannot enter into urea cycle, but it is all available to be converted into orotic acid, causing orotic aciduria. The second type of minor orotic aciduria is associated with the deficient urea cycle enzyme. For example, if ornithine transcarbamylase enzyme is deficient in mitochondria, then urea cycle becomes stopped and it results into the high level of carbamoyl phosphate in the cytosol. When there is more availability of carbamoyl phosphate, there is increased pyrimidine synthesis, which may result into the increased concentration of orotic acid in urine, causing orotic aciduria. But the type of orotic aciduria that is seen in this type of disorder is of mild type. But this mild orotic aciduria can be transformed into severe orotic aciduria if the patient is consuming high nitrogen containing food items, because high nitrogen containing foods may result into the substrates production for urea cycle, and if there is no urea cycle happening, it may result into more production of carbamoyl phosphate in the cytosol, ultimately causing orotic acid. Now the third minor type of orotic aciduria is drug-induced orotic aciduria. There are two drugs which are reported to induce orotic aciduria. The very first one, as all of you know, is allopurinol. Allopurinol is used to treat hyperuricemia and gout, but it may cause orotic aciduria as well as a side effect. Allopurinol may inhibit OMP decarboxylase enzyme. So as you know that if OMP decarboxylase is defective, then OMP cannot be converted into UMP, as a result of which there is high level of OMP, which may result into oretidinuria. And ultimately there will be high level of orotic acid as well, eventually leading to orotic aciduria. And the second type of drug is 6-azaluridine, which is an antiviral drug. It may also inhibit OMP decarboxylase, and the same clinical manifestations would be seen as we have seen in the case of allopurinol. So OMP would not be converted into UMP and ultimately leading to oretidinuria and orotic aciduria. Okay, how can we treat orotic aciduria? One therapeutic approach that we have to fix orotic aciduria is to administer uridine to the patient. Now, what is uridine? Uridine basically is a nucleoside which in the body is converted into UMP first by salvage pathway. And in this reaction, it uses kinase and ATP. Then again using ATP and kinase, it is converted into UDP. And UDP is again phosphorylated in the presence of kinase and ATP to UTP. So this UTP that we get finally from the salvage of uridine, this UTP acts as an inhibitor of CPS2. As you can see here, that CPS2 or carbamoyl phosphate synthetase 2 activity can be blocked by UTP. So when this enzyme is blocked here, then we can regulate the synthesis of orotate and we can control the formation of excessive amount of orotate in the body. So we have to regulate the levels of orotate, because if orotate is present in high concentrations, it may result into the production of kidney stones, ultimately leading to the development of urinary obstruction. Salvage of pyrimidine bases. So we can regenerate pyrimidine nucleotides from the turnover of nucleic acids, which can give us pyrimidine bases. So if you have a pyrimidine base, then it may be regenerated back into pyrimidine nucleotide by a single reaction, which is catalyzed by pyrimidine phosphoribosyl transferase. In this reaction, PRPP enters and pyrophosphate leaves, and any pyrimidine base can be converted into its pyrimidine nucleotide. Here I have highlighted the conversion of uracil, which is a pyrimidine base, into UMP, which is a pyrimidine nucleotide. And if you have a nucleoside, then you can also convert that nucleoside into nucleotide by kinase. So for example, if you have uridine, which is a nucleoside, by kinase and ATP, it is converted into UMP. This UMP again undergoes phosphorylation by kinase and ATP and gives you UDP, and ultimately you can get UTP. In the last slide, we have seen this salvage pathway for the production of UTP from uridine for the treatment of hereditary orotic aciduria. Now we are going to see degradation or breakdown of pyrimidines. When purines undergo degradation, their ring remains intact and it does not undergo any cleavage in humans. But for the pyrimidine degradation, pyrimidine ring undergoes cleavage and it gets opened up and resulted into the production of highly soluble products. For example, you can get beta-alanine, beta-amino isobutyric acid, and also you can get ammonia and carbon dioxide. Okay, this is the complete protocol for the degradation and breakdown of pyrimidine. Here is the general sequence for the pyrimidine nucleotide, and these are the specific pyrimidine nucleotides which can also undergo the same reactions and ultimately can give you end products. So we start with CMP, UMP, DCMP and DTMP. CMP and UMP are found in RNA, DCMP and DTMP are found in DNA. Now in the first reaction, what happens, that nucleotides are converted into nucleosides and there is the release of inorganic phosphate and the enzyme is nucleotidase. So from CMP you can get cytidine, UMP is converted to uridine, DCMP is converted into D-cytidine and DTMP is converted into D-thymidine. Now in the next reaction, there is the activity of phosphorylase enzyme and nucleoside is converted into its free base. From cytidine you can get uracil and again you can get uracil from uridine. Now in these two reactions, there is removal of free ammonia and ribose sugar gets also eliminated. While from D-cytidine you can get uracil, from D-thymidine you can get thiamine. And in these two reactions, there is removal of deoxyribose. Now in the third reaction, free bases undergo reduction and this is done by dehydrogenase enzyme and NADPH is used as a source of protons. So after reduction, uracil is converted into dihydrouracil. This uracil is also converted into dihydrouracil. This arrow here is mistakenly placed and this thiamine is converted into dihydrothiamine. Now in the fourth reaction, there is opening of ring and this is done with the help of hydropyrimidine hydrase. So in this reaction, dihydrouracil is converted into beta-uridopropanoic acid and dihydrothiamine is converted into beta-uridoisobutyric acid. Now in the last reaction there is simple hydrolysis, which is carried out by hydrolase enzyme, and you can get your desired end product. So from the hydrolysis of beta-uridopropanoic acid, you can get carbon dioxide, ammonia and beta-alanine, and by hydrolyzing beta-uridoisobutyric acid, you can get carbon dioxide, ammonia and beta-amino isobutyrate. So beta-amino isobutyrate, beta-alanine, carbon dioxide and ammonia are the end products that we get after degrading pyrimidines. Last part of today's lecture is focused on studying some natural and synthetic analogues or derivatives that can be obtained from purines and pyrimidines and their significance or importance in health and disease. So first are some natural derivatives. As you know, that cyclic AMP and cyclic GMP are important cyclic nucleotides which are involved in signal transduction cascades. They act as second messengers and they mediate several cellular responses in the body. Then there are some activated forms of carbohydrate and lipids that contain nucleotide as important part in their structures. A few examples are UDP glucose, as you know that this is involved in the synthesis of glycogen. Then UDP galactose, which is involved in the biosynthesis of polysaccharides. And then we have CDP choline and CDP ethanolamine. These are the compounds which are used in the biosynthesis of phospholipids. Nucleotides in the form of vitamins may act as coenzymes, and you are much familiar with these examples like NAD, NADP, FAD. These are involved in oxidation reduction reactions and in many metabolic pathways. Then there are some derivatives which are important in catalyzing transmethylation reactions where there is a need of transfer of methyl group from one to another. For example, acetinosyl-methionine, which is also called as SAM, and this acetinosyl-methionine is involved in number of reactions that are involved in protein metabolism. Then there are some forms of purine and pyrimidine derivatives which act as regulators of multiple number of metabolic events in the body, and they can increase the levels of cyclic AMP. Notable examples are theobromine, which is 3,7-dimethylxanthine. It's a derivative of purine, and this is found in cocoa. And then we have caffeine, which is 1,3,7-trimethylxanthine. This is also found in coffee. So these two are important examples that can boost cyclic AMP levels in the body. Synthetic or man-made analogues, derivatives or drugs. These are widely used for treating cancer and some other infectious diseases. Now what is their underlying mode of action? Basically, we take natural purine or pyrimidine and we alter its ring structure of the sugar moiety. And it is modified in such a way that now it can induce toxic effects on the target cells. So what these synthetic analogues can do? Basically, they can inhibit the activity of specific enzymes which are responsible for making nucleic acids in the cells. So when nucleic acid synthesis would be impaired, then cancer cells won't grow. Similarly, there is another mode of action as well, that these synthetic analogues can also affect the base pairing process of DNA. And this base pairing process, when it is interrupted, then there is inaccurate transfer of genetic information and cancer cells cannot divide or disseminate. Okay, some important examples of synthetic analogues. First is 6-thioguanine. This is a synthetic analog of purine in which at 6th position thio or sulfhydryl group is added, and now it becomes modified and used for treating cancer. Then we have 6-mercaptopurine. This is also synthetic analog of purine in which again sulfur group is added, and it becomes altered or modified in such a way that it can be used for cancer therapy. It can also be used to treat some autoimmune disorders. Then azacitidine and azaguanine. Azacitidine is obtained from cytidine by adding nitrogens to it, and then azaguanine, it is a synthetic analog of purine in which nitrogen is added. So these are widely used again for treating many types of cancer. Then we have allopurinol, which is a synthetic analog of hypoxanthine, and this is used for the treatment of hyperuricemia and gout. Then we have cytarabine, which is also called arabinosyl cytosine. This is also shortly abbreviated as ARA-C, and vidarabine, which is also called as arabinosyl adenine and ARA-A. So in these synthetic analogs, basically arabinose sugar is added in place of ribose, and their structures become modified, and these are also used for treating various types of cancers. Here is another example of synthetic analog, and this is azathioprine, in which nitrogen and sulfur both are added, and it is used as an immunosuppressive agent. It prevents graft rejection in organ transplantation. Then we have 5-iodo-2-deoxyuridine, which is used for the treatment of herpetic keratitis. This is basically an infection of the cornea which is caused by herpes simplex virus. Then we have 5-fluorouracil. This is an anti-cancer, anti-neoplastic or antiviral agent which is used to treat several types of cancer. Okay students, activity for you. You are supposed to recall today's lecture and give the answers of these two queries. I'm going to reveal the answers. If you have answered both queries appropriately, it means you have understood the all concepts of today's lecture.